So our speaker today is our very own Dr. Wendy, and she's going to talk about metals and metalloenzymes. So the research program that she heads is, I think generally it's plant physiology and genetics, but there's a lot more to it than that. A lot of really cool techniques like omics, and cellular imaging are going on. Like I've worked with Wendy, collaborating on proposals, and I'm also on graduate committees with her, and that's the experience I've had. She got her PhD from the University of California, Santa Cruz, and I think 2 bachelor's degrees, one in biology and one in chemistry, with honors from the California State University of Bakersfield. So with that.
Oh, thank you so much. And thank you all for this opportunity. I'm really excited to tell you about some of the work in my lab. For our really broad audience, which is fantastic, one of our strengths in the department, I decided that this would be of the greatest general interest. So that means I'm not talking about one of the near and dear to my heart are these specialized compounds and how they interact with in the plant cell itself, and also how it mediates communication and processes in other organisms. So we'll do the metalloenzymes. So because something related to some of my disability is that sometimes I don't speak so clearly or so loudly, so please raise your hand if that's the case, and I really welcome your questions and to talk about things. And I'll also try not to geek out too much. So if I am geeking out too much, especially near the end, because I get really excited and I talk really fast and things, so again, please, raise your hand, let me know, and make it clear, make some adjustments. All right, so that's so big. I decided to focus on 3 major areas for this presentation, let you guys know what's going on. First is an update about this iron binding peptide and the high iron and high zinc rice. So that's pretty cool. Second is, everything old is new again. Metal accumulators. Yeah, I didn't think I would be back working on metals again, but I am, and you'll hear about that. And then finally, a couple things about metalloenzymes. So I'm talking about some programs and some collaborations, and then the big resubmitted paper. Yes. So okay. So something that's always been important to me is feeding the world. Ever since I was a little girl, and there was starting people in China, I wanted to send them my dinner in a box. I was a little girl. I didn't know that wouldn't work. But here's a fact that I don't know. How many of you are aware that 25% of pregnant women die because of iron deficiency? Yeah, this is a really big problem, obviously. And one way to address this is with some iron fortified rice. So this rice not only can accumulate iron, it can accumulate zinc, and zinc is also important to prevent stunting of stunted growth in children. And this rice is really cool too because the grain weight is more and the grain yield is more, so it's also attractive to farmers to grow. And all it took to do this was to use this 9 amino acid iron binding peptide, which discovered way back in Santa Cruz. So this has been going on a long time, trying to see it. We use this, kind of snuck this in with a biofuels project to make some next generation drop-in biofuels. And it worked with the grains, which is really great. And so you think, well, it's great. Shouldn't this be out there? It's like, well, obviously there's a problem. One problem is it was transgenic, and we've been hearing from, even back in the mid-2000s, oh no, we can never put transgenics in the field, especially in Africa, it won't be accepted. And the other problems with societal acceptance of transgenics, and the EU put out a guideline for however long ago it was that if it's transgenic, it can only have less than 20 base pairs, which equals about 5 amino acids, which in reality is like no transgenics. It's just a sneaky way to say it. So this is really only 9 amino acids, so it's totally not viable. But with the advent of CRISPR editing, specifically a new prime editing from Dr. Yiping Qi, who happens to be just down the hall from me, and with the fully sequenced rice genome, we can overcome this with this gene editing. So I was giving a presentation before, and Yiping heard me talk about this rice and said, 25% of women are dying. This is big. We have to do something. So he wanted to collaborate with me on this. And because we have the rice genome, I was looking for the rice sequence that we could modify. And so I found one, and we just need to put on these 2 amino acids, which are these 6 base pairs. And so we're doing it right now. So after we get the rice and bulk it up and do the field trials to see if it's really efficacious, then the International Rice Institute said that they wanted it to interbreed with their elite line. So we have a path forward for it, for getting this high iron, high zinc rice out into the field or into people's tummies. Okay, so that's my update on that. The second topic is on metal accumulators. So we can kind of divide it into 3 different kinds. So there's metal sensitive, which is fairly obvious and doesn't really need a definition. And then we can have metal tolerant species. They can be tolerant in one of 2 ways. One is they can totally exclude the metals from entering the system completely. And the other ones can accumulate it, but then they can store it in say, an organ like the vacuole. And that way, it's not going to be damaging to any of the cellular processes, because a lot of the metals can undergo Fenton reactions, make reactive oxygen species. They can also poison the different enzymes in the cell by binding to them when a different metal is the enzyme that's needed for it to function properly. So, of course, the cool thing about plant roots is this very root tip region here is completely continuous with the rhizosphere. So any water or metals can just come in right on from the root tip, and that's the apoplastic pathway. Otherwise, they can come in through the symplastic pathway, so through transporters that takes them into the cells. Then they can be transported throughout the plant, and some, like selenium, can be conjugated to amino acids and then released as volatiles into the environment. And then the third category are the metal hyperaccumulators, and then I've listed the definitions of how much they have to accumulate in order to be considered hyperaccumulators. One thing I didn't put on the slide are indicator species, which generally accumulate amount of metal proportional to the metal that is found in the environment. And so those were initially thought to use for bioprospecting. So, like I said, everything old is new again. I decided to put this one in here because we are wrapping up some of this older work there's some ecotypes that we're collecting in calamine and serpentine soils. And there's also this Arabidopsis lyrata that was found at a mine site not far from here in Friedensville, Pennsylvania. So there's a couple populations. There's one at the zinc mine site, which accumulates zinc. This one in the wood site, and the other Arabis species are not metal tolerant or accumulate. So why, you can see here's the plant growing in a 4-inch pot. You can see it's really not very good for phytoremediation. And most of these plants that are hyperaccumulators don't have a lot of biomass. So for any kind of remediation, I think it has to be full bioremediation. You have to have the soil microbes and other factors involved, because these plants are just too small. Any species, philosophy, anything. And we'll get this published. This is old data from Santa Cruz when I fell in love with signals between plants and fungus. And here's a Arabidopsis lyrata. Arabidopsis isn't supposed to form any, Thaliana anyway, any rhizo associations, but this one does, and it also accumulates a lot of cadmium as well, and we compared it to an arenosa that was collected in a zinc mine site in Switzerland, and so that's some things. Of course, cadmium is very bad. Okay, so on here's the everything old is new again. The nickel mining, phytomining, has come back. It's not ecologically friendly at all, obviously. So when the DOE came out with the ARPA-E for phytomining, Rufus Cheney, who just lives in Beltsville down the street, called me up and said, "Hey, Wendy, we've got to do this." I was like, "Oh, do I really want to do the metal work again?" But anyway, he convinced me. His Alyssa Morale, it's been renamed because of the invasive problems in Oregon that Stephanie is probably aware of as well. But thinking about it and how horrible regular mining is, that you're thinking about how we can get this to work. And so we spent almost a year getting the preliminary data for the grant proposal, screened several different species. We chose Chalcidica in the end, and Angus was screening them, and he found one that grew really fast, that bloomed late, and so we have a near isogenic line of that we can use for that. So how are we going to make this minor invasive species not invasive and to actually work? Well, we already just with those some cultural conditions got halfway to what the DOE threshold was that we're supposed to get to. So our plan was to get the rest of the way by, again, using CRISPR editing, so it's not transgenic, and we can be very precise with prime editing as opposed to the original CRISPR, which was very messy and can end up with different deletions and insertions and such. So one way is to increase the amount of biomass, to extend the vegetative growth cycle, and to suppress seed production dispersal. And that's in collaboration with Dr. Daniel Rodriguez, who is a new faculty member in plant sciences. So he's actually just up the stairs for me. So do you guys want to know any of these details? Okay. And so, come to the third topic, and I'm going to talk about this PPIase activity. I have a couple of these peptidyl prolyl isomerases, and I'm showing you this one because it's a new collaboration that I have with Chris Clark over at USDA. So what is this peptidyl prolyl isomerase? Well, it's called a cyclophilin. It's a corrector protein. So it's most famous for cystic fibrosis. If you don't have this, then you're going to have the wrong, if it doesn't have one that's a corrector for the proteins, then that causes the terrible problems with the lungs. So there is a homologue in plants that is similar. It's twisted dwarf 1. So what a corrector does is if there are two prolines next to each other, or the proline's in the wrong direction, then it can just switch it. So it makes sure that proteins are in the correct conformation so that they can have the correct function and activity. So it can fold and refold. It's also involved with calcium signaling and plant immunity. And if we look at the little twisted dwarf, it's so tiny, it's so cute. It's this little tiny cabbage that's very, very twisted. And so here's our crystal structure I was just showing. So if we delete the C terminus, just cut this part off, then the plant grows really, really big. It flowers earlier. So when we're looking at targets for twisted dwarf and how it can function, one was to use the bacterial toxin called the Faxtamin A. And as you can see, the poor little mutant is very sensitive to it. What I don't have here is a picture of this twisted dwarf minus the C terminus, which is completely resistant to thaxtamin A. So you'll have to believe me because the picture's not there. Anyway, so as I mentioned, it's a bacterial toxin. It's made by Streptomyces species, and it's the cause of potato common scab. Its functions are for inhibiting cellulose synthase. So say inhibits cell division, it promotes programmed cell death, and calcium signaling. So you can see some overlap with some of the functions that we know about twisted dwarf. And these potatoes actually happen to be from our garden from the summer, and here's some scab. It's mostly just on the epidermis, on the peel, and they make these bumps. Sometimes they look really severe. They make it so that they're not sellable. People, they really can't sell them. People don't want to buy them in the store. Sometimes they are used for potato chips. You can see, you check your potato chips, you can watch the edge, you can see that. This is, some of the species can actually infect the cortex, and you can see this kind of pattern in there. So it's really damaging to potato crops. It happens at a very, very particular small, tiny window during tuber development of the potatoes. So it's really hard to put your finger on the mechanism and when it gets infected. So after one of the plant biology symposiums, I don't know, it just came up talking with Chris. He was talking about thaxtamin A, and was like, oh, twisted dwarf. And so we started a collaboration. We found some other candidate genes as well that are also expressed in the potato epidermis. And so we can edit those and look at that. And we'll have a joint postdoc coming soon. So we're also really excited to get this off the ground. Okay, do you guys have any questions so far? Okay, all righty. So this is the last topic, is looking at the dioxygenase for auxin oxidation, or DAO. I'll just call it DAO. And so this work was more than 500 independent experiments, so it's really huge. Some of them were repeated independently by different people in the group, and some were also repeated blind, because sometimes we want to make sure there's no bias, and oh, I expected that, and it was so some of them were also done blind. And guess what? I'm going to tell you about every 500. No, I'm joking. I'm not going to tell you all 500 experiments. But the three main things we wanted to address in this study is to nail down what is, or try to nail down, what is this membrane-associated auxin oxidase activity, because in the literature, from 50 or more years ago, there's the soluble oxidation activity and there's the membrane activity. And we're pretty sure that, the soluble activity is DAO, but the outstanding question really is the membrane activity. The Brassicacea have a second copy of DAO. A few other species may as well. It looks like rice might have a second copy, so that's very important. So what is it? Is it just redundant, or does it do something special? And you can think, if it's redundant because the genes are so close together, they're completely linked. So can't do genetics with them independently. But we have a cure for that. And then the final question was, does this oxidized auxin, oxIAA, have a physiological function? Because there's 5 to 10 times more oxIAA than IAA auxin in the cell. So is there something we need to know, something that we're missing? So we set out to answer those questions. So here is some data which I think support that DAO1 fused to YFP, yellow fluorescent protein, is membrane associated. And there's some other groups who have similar data. So we have lively discussions. I'm particularly proud of this picture because I took it myself. But we have had lively discussions, Jane and I, about whether or not it was membrane or soluble. She was convinced it was only soluble. So graduate students, it's really good to have these healthy discussions with your PIs, and it's okay to disagree. So, Mark was like, yeah, that looks like it's on the membrane. So Mark was like, I want to know for sure. So he just took this on all by himself, without direction. She's like, when do you want to do this? I'm like, sure, yeah, go for it. So he did. So here, if you take plants and you grind them up, you can separate them into the soluble fraction and a membrane fraction. So we can look at this and we run the gel and they do a Western blot. So we use an antibody against YFP. And you can see that Dow is in both soluble, S soluble, and M membrane fractions. Maybe there's a little bit more in the membrane fraction. And then the next question is, well, that's great, it's on the membrane, but we can do more. It's like, what membranes can it be on? So you can take these microsomal membranes and then run them on a sucrose density gradient. So you put the microsomes on top and you have a sucrose gradient. That's actually kind of fun to pour. Maybe we can get someone to pour us a gradient over at Looney's. I can show you how it really works. So, and then you can run the gel and do the Western blot. And so Dow 1, as you can see, is mostly associated with the plasma membrane and the endoplasmic reticulum. So Mark had this really great idea. He wanted to treat the membranes and the soluble fractions with auxin to see what would happen to Dow 1. Where does it go? Does it affect anything at the membrane? So here's our control again with the soluble and the membrane fraction. He treated with either 100 nanomolar IAA or 1 micromolar IAA. And so what he saw was a shift from the microsomal fraction to the soluble fraction in both cases. So that tells us that we know that its membrane association is dependent on auxin. So if there's a lot of auxin that is coming into the cell through transport, Dow is getting knocked off. This is hypothesis, haven't shown it yet, getting knocked off the membrane. So it's not going to be there to oxidize it. So the auxin is available to travel to the nucleus and do its job of auxin signaling and turning on gene expression. Still have to. Of course, the assumption was there is that it's doing it on the membrane. So that's what I thought. There's no room to draw, so good idea. Okay, so in order to understand about its enzyme activity, we have to know a little bit about what the pathway is. So auxin can be made from tryptophan and amino acid in 2 steps. And so we know that we thought that Dow was just directly oxidizing auxin, and that after the Gretchen-Hagen 3 or GH3 proteins conjugated auxin to amino acids, that Dow could oxidize these and take them out of the system. But one of our colleagues, Ken Hayashi in Japan, did some extra experiments. He had these really cool mutants and did an extra set of experiments where he showed that these imidahydrolases, which are resident within the ER, and so they're inside of the ER, could cleave the amino acids off of this OXIA, and that OXIA was this is the main pathway for oxidation. So that still leaves the question, can Dow directly oxidize auxin? Because this all happens in the soluble fraction, and we were interested about the membrane fraction. So I've just highlighted some of the data, and of course, I'm not showing you like 5 other tables. But so if we look at the soluble fraction again, which we know there's some in Dow, and we can feed Columbia 0, which is the wild type, some IAA, and then yes, we see auxin as we expected, and give it the IAA asp. And yes, we get OXIA made. It's wild type. It has all of the proteins needed to do that. Then we took the microsomal fractions. we did the same thing. So the microsomal fractions were, all these experiments were optimized for DAO and not optimized for these soluble proteins. Even here, these add-backs were soluble proteins from the mutant spent. You can see that giving them the substrates, the membranes, and seeing what happens, that yes, we are still getting some OXIA production, and that leads us to conclude that yes, DAO1 is capable, or DAO is capable of in the membranes of directly oxidizing auxin. It doesn't mean it's the only one, but it's no one has been looking for this for a long time, and it can do this. We can't directly compare these numbers either about, oh, it's half the activity or half the auxin is oxidized, because that's beyond the scope of what these in vitro enzyme assays can do. But so we showed that. So the second copy of DAO, so a second part of looking at DAO. So we have these two genes, DAO1 and DAO2. This is DAO1. We had this transgenic a long time ago, and then Ruben was making these CRISPR lines. And so we have these point mutations, and this one has a big deletion, because we were using this really old-fashioned, so old-fashioned now, CRISPR that could also take out some deletions. And the reason why there are 3 here is because those are different gene models. So different messenger RNAs were noted when people were assembling that and comparing it to the genome. And so they have different amounts of 5' or 3' untranslated regions. And sorry, that's like way too geeky. Okay, but the reason why we had to use CRISPR is because they are so close together that their chances of crossing over, remember good old Mendelian genetics with the P's and the, that would never happen. Or it could, like in 100,000 crosses or something, but they're just a few hundred base pairs apart. So now that we have these, we can look at the single mutants and the double mutants and look at their metabolic profiles to see what happens. So we can look, here's the wild type Columbia, DAO1, below the limit of detection, DAO2, and then the doubles. So I'm just showing you the oxide in the shoots and roots that we separate from each other from some 7-day-old like Rome seedlings. And so, so you can see that DAO1 is the primary auxin oxidase. We can see that the activity in DAO2 for the OXIA accumulated is the activity of DAO1 by itself. And basically the double mutants all look like the DAO1 mutant. But if we look in the roots, and we look at, here you have Columbia, the wild type, and we look at how much OXIA there is in the root, there's more than in the wild type, which was a really big surprise. So we checked to see, well, maybe DAO1 is upregulated so that there's more protein, that's why there's more OXIA. But it turns out that's not the case, and that the hypothesis is there must be some post-translational regulation of DAO1 that is compensating for the loss of DAO2 in the roots. And we can say, well, and for fun, we did some Orbitrap with Rhett Cooper. This is part of some work that Liliana did for her thesis. She's now in charge of the exosome facility, so anyone who wants to run PFAS is what she's mostly doing at the moment. There's a big PFAS project there, so she's doing all of that. But it's really cool with the Orbitrap. So usually this is, we analyze by using LC mass spec, and that's because we know what we're looking for and we want to quantitate it. Orbitrap is really cool if you, it's completely untargeted, so if you don't know what you're looking for, it's really great to do this kind of mass spec. And so we found, comparing DAO1 to Columbia, 7 different significant compounds, 13 in DAO2, but 101 in the double mutant. And so there's some interesting things in there to look at to see what's going on there. So we saw with the metabolites that the roots were different with DAO2. And if we look at spatiotemporal expression, looking at protein abundance and transcript abundance, we can compare them. And then we can also just look at relative protein amounts and compare DAO1 and DAO2. You can see that DAO2 is mostly expressed in the hypocaudal and in the root. And we can do some phenotyping, look at some phenomics, and see how these are behaving with looking at different parameters. So the DAO2 mutants have longer hypocaudals, have longer primary roots, and so that's consistent with where its major expression domains are. And also it has slower germination rates. They eventually germinate. They're still like about 4 days late. So any experiments with them have to be timed and staggered. Also, that's with the fresh seeds, but also if you have after ripened seeds, it seems to rescue that part of the phenotype. And if we look at the double mutants, they have more lateral roots and more adventitious roots. They also are taller and flower faster. Now, I did, for these double mutants here, the primary root length, because if we look at DAO1, it's similar to wild type, but the double mutants are similar to DAO2. So this would be, we can attribute that to the function of DAO2 and not to DAO1, whereas most of the other phenotypes of the double mutants are really based on the activity of DAO1. So DAO2 is functioning in a very smaller domain and subset of physiological responses. So another cool thing, of course, plants can't run away. They have to respond to their environment. And so there's some things that we know that it's important for. It's important for gravity responses. They actually bend faster. For phototropism, so bending to light, that's also faster. For photomorphogenesis, their apical hooks open faster. And this is all circadian, and also the oxide synthesis is circadian regulated. So you can watch it over the course of the day go up and go down. And so the apical hook is on the next slide in temperature. Well, get to the apical hook in a minute then. So the last one is temperature. So when we brought the cart up upstairs from the growth room and we're looking at the flowers to do some phenotyping, and it's just like, whoa, that plant has 5 petals. Okay, Arabidopsis only ever has 4 petals. It's like, that has 5 petals. And everyone who'd walk in the lab like, yeah, it's 5 petals. It's 5 petals. Even the brand new shiny undergraduate, oh, that has 5 petals. So, everybody was, but nailing it down that it was temperature was really hard because we was thinking, oh, it's day length, it's circadian, it's something else. So it took a really long time to nail down that this was related to temperature. And wild type will occasionally have some, so it's not like unheard of, and a temperature shift can do it. So it's basically so they have some basal level, but then that's a lot of flowers that people counted. So you did a 12-hour shift from 22 degrees to 26 degrees and waited 9 or 10 days until the new petals came out to count them. And then, yes, there's significantly more aberrant petal numbers than that. So how can that be? Well, part of it is this. It's the third and last section of this talk, and that has to do with auxin signaling. So how does auxin signaling work? Well, here's a gene, and there's a transcription factor, an auxin responsive factor, and it's the oxia proteins. Yes, unfortunate name, but that's what they're called. OXIA proteins sit on top of it and repress gene expression. That's what the little X is. Now, the auxin receptor complex is actually a co-receptor complex where the IAA acts as a molecular glue. That is the technical term in the paper, the molecular glue, between the tier 1 and the OXIA repressor protein. So it brings them together in a special way, and there are 2 prolines involved, and that's my other one I'm not talking to you about today, but it brings them together, and then the OXIA repressor protein gets ubiquitinated and degraded. So after it's gone, then of course that dissociates, and then gene repression is released. So when we look at auxin signaling, we can use an auxin reporter as a proxy to see where the auxin is and where the auxin activity is happening. So when it's called DR5, it actually comes from those GH3 proteins I talked to you before, a little bit of that promoter. And so we can use GUS or we can use GFP. GUS makes this blue precipitate. We can use GFP, and we can see that looking at this very special apical hook, which protects the meristem of the seedling as it grows up through the soil. So there's a real purpose. After it reaches the soil, then it can see the light and open up. And you can see that Dow has more signal in the apical hook than the wild type does. And we can also quantitate the amount of auxin, OXIA, whatever metabolites we want to look at in this apical hook upper hypocaudal region. These are really tiny, so you really can't cut those off, and so some of the apical hook is there. So if we just concentrate on the Columbia and look at auxin, we can see that over time from 48 hours to 70 hours, the auxin is increasing from about 20 to about 40. And we can look at the OXIA as well. That is increasing from, say, about 100 to 140. But the thing is that the auxin to OXIA ratio is decreasing over time. So as we're getting more auxin, we're getting more auxin faster than the OXIA. So the nice thing that we're able to do is do some dynamic analysis. So mostly when people are looking at these apical hooks and looking at phototropism and other aspects of development, it's just a static time point. And we were able to start to doing a dynamic assay to see what's going on in over time in this way. So if we just look at 2 days versus 3 days, we have Columbia, it's not really opening very much. You can see in Dow that the apical hook is more relaxed, and definitely by 3 days, it's more open than the wild type. And we can also take the wild type and treat it with different, a dose response curve essentially of auxin, and you can see that auxin is inducing hook opening with the amount of auxin. And if we look in this yucca-6 mutant, you might remember that, auxin two-step production. So yucca-6 is an auxin overexpressor. It's a dominant mutation where it makes more auxin. And so even in exogenously applied auxin to this, if you could imagine this just right there with a little Hamilton syringe, a little just drop of auxin there. But here we have an endogenous one where the apical hook is opening faster with auxin. So we know that auxin induces hook opening. What does OXIA do? So, it delays hook opening. So we can look at what happens with Columbia. So here's, at 2 days, we add OXIA, the hooks, and we can add it there. It seems a little tighter, less variation. However, at 3 days, because it's also making more auxin, the OXIA is not significantly different. We can also look at the wild type here as another way to look at it, where we have the mock treatment and treating it with OXIA, where it's keeping the hook angle, we add auxin, and it's opening very quickly. If we look at the Dow mutant, we add OXIA to it, then it phenocopies the Columbia, so the hook becomes tighter than it was, and we see some effect at 3 days as well. And if we look at it with this kind of assay, you can see that the gray, which is the oxi-A, helps it to phenocopy exactly the wild type curves. So oxi-A can rescue that phenotype from DAO1. And so this is my last data that I'm going to show you today. And this is a bioassay also for auxin signaling. So this first one is the biolayer interferometry, where tier 1 and IAA7, one of the oxi-A proteins, are in this assay with 200 micromolar oxi-A, and as you can see with using the different concentrations of tier 1, that we're able to determine the dissociation constant for oxi-A binding tier 1. And we also did some yeast assays as well. These are assays with yeast. And here's this YFP control, so it's not degraded, but in this control with the different amounts of auxin and oxi-A, then you can watch the rates of YFP going away, its fluorescence going away. And so these data tell us that oxi-A has 10 times less affinity for the auxin co-receptors than IAA itself, which is good because otherwise there wouldn't be any auxin signaling, and auxin can outcompete the IAA. So this means when auxin is low, when the etiolated seedling in the dark is younger, then oxi-A is binding the co-receptors, and it's preventing the degradation of the oxi-A protein, and that's why we're keeping the hooks tight. But as the seedling is aging, or if it's expressed to light, the auxin concentrations are going up. Higher auxin concentrations displace the oxi-A, and then we have resumption of normal growth. So from this kind of stasis part where the seedling is waiting for signals to grow, it has to get above the soil and whatnot. So it's to basically keep it safe, help for seedling establishment. Yeah, okay, so here at just my conclusions, yes, DAO1 is membrane associated. Yes, it can directly oxidize auxin. It's able to. DAO2 does have some distinct functions from DAO1. They both modulate different physiological responses, both developmental, abiotic, and integrate them. And oxi does have a physiological function during auxin signaling. We found one instance, one bioassay of the apical hook, and there are likely others. And these are everyone that I'd like to thank who are involved in the paper. And thank you for your kind attention.
I think we have time for a couple quick questions. Dr. Yarrow?
In the middle of your presentation, you talked about nickel, and you talked about bioaccumulating plants. And
Yes.
you mentioned that basically most hyperaccumulators, really from a applied perspective, they don't produce enough biomass. Is there work to try to create transgenic bioaccumulators? Is that a possibility?
Yes, that's a really great question. So and thank you. Which I skipped a part. These the Odorantharina, the Lissamorelli, that can come about this high. So that one has the biomass. It's specific for nickel. It's actually being used right now in like Slovakia or neighboring countries for nickel mining. We're just considering it now in the U.S. We'll find out in November if that grant is funded to work on that. But most of the brassicaceae are tiny. There are trees in New Zealand that bleed green with nickel if you cut them. Yes.
Oh wow.
I know, they're really cool.
Crazy.
It's really cool. But yeah, it's just I have transformed like all of these brassicacea already, so I know we can do, that's why I know I can do the Alyssum project, but from work before. So if it blooms, I can transform it, and if it doesn't, there are new methods in tissue culture to do it. But to really beef up the biomass, well, I would just use the same playbook that I already showed you for how to do it, but to get them like tall enough to mow it, because you can't mow a rosette plant.
Right.
So it has to be a different kind of plant. Yeah, unfortunately, most of them are brassicacea, and they're rosette plants, except the alyssum, this weird alyssum that's
What about canola?
yeah.
Because canola could be, I mean,
That's
canola-
big, yeah.
That's big. I'm just curious.
That's great. I didn't think of canola. Well, so it's probably a lot of gene cassettes that need to be put in
Yeah.
so that it's not toxic to the plant. Is it doable? Probably. How long would everything takes time and money, but other than hands. So.
Right, yeah. No, thanks.
No, that's great. So maybe it is possible for bioremediation. Thank you.
Yes, I think we'll have to stop it there, so we don't get pushed out by the next class. But if you're interested to talk to Wendy more, then join us at Wendy's.
Oh, thank you.